Hello, my name is Maribel Gendreau, and I teach research methods at Hampton Academy, a K through 12 independent school in Newport News, Virginia. The class is an inquiry-based two-semester science elective, offering high school students the opportunity to research a topic of interest in science and technology. Students are encouraged to investigate real-world topics and design, plan, and implement a semester-long investigation to address a research question. During the first semester, the course explored various topics such as research methodology, ethical research practices, biotechnology, and data analysis tools. The second semester, students work towards the completion of a research project, culminating in a final research paper and oral poster presentation. After the course, students will have the option of taking the biotechnology assistant certification exam. In this course, students learn basic skills essential to conducting biotechnology research. They're introduced to microbiological and cell culturing techniques, as well as DNA and basic protein analysis techniques. They also learn how to keep an accurate account of their experiment in their lab notebook. By the end of the course, students understand different scientific research designs and models. They learn how to set up a research study, perform literature reviews, and refer to and cite from scientific literature. Each quarter, students select a topic in science affecting the US or world population, review the literature, and write a persuasive paper expressing their opinion, which they will defend in a Socratic seminar-style presentation. In addition, they read A Cracking Creation, Gene Editing and the Unthinkable Power to Control Evolution, and reflects on the topics learned through discussion boards. Anna is an 11th grader interested in pursuing environmental science in college. Her research idea originated from a talk at a commodity classic in Florida.
Hello, my name is Krzyzanowski and I'm a junior at Hampton Academy. For my investigation, I studied the effects of the addition of amino acids had on the growth of corn, wheat, soybeans, bell peppers, and tomatoes. The central finding of this investigation is that the addition of amino acids did not affect the morphology of the soil bacteria colonies or the growth of the plants. The graphs below represent the plant height and number of leaves grown. As you can see in the graphs, the addition of the amino acids did not lead to a significant difference in the height. Day 15 and day 30 plates are below the graphs, and they show that there was a consistent presence of similar bacteria in all the plates with size and color changes similar to that of the control. This implies that the addition of amino acids did not affect the type of soil bacteria present. Across the globe, there are currently over 870 million people facing food insecurity, and this number is set to double by 2050. Because of this, new ways to quickly produce foods while still protecting the earth are necessary. This investigation aims to find a way to make plants grow quicker while still protecting the earth. It was hypothesized that the plants receiving high doses of amino acids would grow taller and produce more diverse bacteria. In this investigation, corn, bell peppers, tomatoes, wheat, and soybeans were grown in soil in a self-watering container. A high and low dose of amino acids were added to the plant soil, with a control plant receiving no amino acids. The plant height and number of leaves were measured twice a week. After 15 days, soil samples were collected from each of the plants, and a 5-fold dilution was performed on all samples. The 10 to the 3 and 10 to the 5 dilutions were plated and observed for individual colony morphology. It was found that the high-dose plants, except in the corn, emerged later than the control in the low-dose plants. It was also found that there was no significant difference in the height between the control and low-dose plants. With respect to the number of leaves, the tomatoes and soybeans produced an extra leaf in the high-dose plant, while the opposite was observed in the pepper and wheat plants, and all the corn plants had the same number of blades. Across all the day 15 plates, there were dark yellow circle colonies, and these colonies were not present in the day 30 plates, and instead bacteria showed as punctiform white colonies. This investigation did not support the hypothesis that the high-dose plants would grow taller, and this is because they emerged later than all the other plants. The addition of amino acids are supposed to help speed up plant growth, but it actually did the opposite in this case. It is likely that too many amino acids were added, which hindered the plant growth. So in regard to the soil bacteria, there was no difference in the controlled, low, and high dose, the morphology of the soil bacteria present in all the plants. The plate showed as the same morphological changes throughout the Days compared to the control plate, the day 15 plate showed circular yellow bacteria replaced by punctiform white colonies by day 30. This implies that there is likely no direct correlation between the type of bacteria present and the addition of peptides. This change in soil bacteria is assumed to be because of the symbiotic relationship between the soil bacteria and the plant, as different types of bacteria may be needed throughout different developmental stages. In regard to plant height, there was no difference between the control, low, and high dose in any of the plants. Current literature supports that amino acids should make plants emerge faster because they increase energy source and improve photosynthesis rates and chlorophyll biosynthesis. Further studies should focus on the growth of bacteria using different concentrations of amino acids, as well as to determine the effects adding amino acids to plants under stress. In addition to a more detailed study could be conducted to see if there are any differences in the amount of bacteria produced by amino acid treated plants.